So let us now see about the genetic polymorphism in proteins. Genetic polymorphism in milk proteins. So this is we are discussing about the milk proteins. Okay. Now in 1956, R. Skaffenberg and J. Dewey are the two scientists who discovered that beta-lactoglobulin exists in two groups A and B, which differs from each other by only one amino acid. So that is the qualitative difference. That is alanine, valine at position 118 in beta-lactoglobulin and B respectively. So in beta-lactoglobulin A we have alanine, and in beta-lactoglobulin B we have the valine at position 118. So this is known as quantitative differences. Now milk of any food-inducible animal may contain beta-lactoglobulin A or beta-lactoglobulin or both, and the milk is indicated as AA, BB or AB with respect to beta-lactoglobulin. So milk of any food-inducible animal may contain beta-lactoglobulin A or B or both, and the milk is indicated as AA, BB or AB with respect to beta-lactoglobulin. This phenomenon is referred to as genetic polymorphism and has been shown to occur in all milk proteins. So this phenomenon has been shown to occur in all milk proteins. So a total of 60 variants of casein and whey proteins have been demonstrated by Page. Total of 60 variants have been of casein and whey proteins have been demonstrated by Page. So therefore it is likely that more than 60 polyforms exist, and genetic variant present is indicated by Latin letters, that is if A, B, B or these are indicated, or sometimes there are superscript also there, like A1, A2. So these are indicated by upper letter, Arabian letter A1, A2, and then sometimes superscripts are also there. Okay, so this phenomenon is known as the genetic polymorphism. Now, what is the significance? Somebody asked, what is the significance of genetic polymorphism? The frequency with which certain genetic variant occur in breed is breed specific. Frequency with which certain genetic variant occurs is breed specific. Hence, genetic polymorphism has been useful in the phylogenetic classification of cattle and other species. So, first significance is that studying about the genetic polymorphism helps in the phylogenetic classification of cattle and other species. Now, also some of the technological important properties of milk proteins, for example, cheese making properties and concentration of the proteins in milk are correlated with the certain polyforms. And significant research is going on this. So the research is going on whether we can correlate some of the technological properties of milk, like cheese making and the concentration of the protein in milk with the polyforms. The studies are still going on. Now, the casein genotype significantly influences the milk yield. It influences the milk yield, fat yield, and protein yield. It has been observed that the casein genotype significantly influences the milk yield, fat yield, protein yield, with the highest yields obtained for the genotype BB. So, milk which is having genotype BB has been reported to have higher milk yield, fat yield, and protein yield. Similarly, cheese yield on a fixed amount of milk and fat yield were significantly related to polymorphism, observed to be beta-lactoglobulin B genotype, and highest estimates has been obtained for BB. So beta-lactoglobulin milk which are having beta-lactoglobulin BB genotype is reported to have higher cheese yield on a fixed amount of milk, and also the fat yield that has been observed. The protein percentage was influenced by alpha S1 casein majorly and kappa casein, with the genotype BC and BB respectively having the highest percentages. So milk having the alpha S1 casein BC and kappa casein BB have been reported to have higher protein percentages. So this is the significance which we are studying. And also milk containing kappa casein B has been shown to exhibit better heat stability. So milk which are having the genotype kappa casein B are shown to have higher better stability. And if we compare the beta-lactoglobulin among beta-lactoglobulin, BB is having higher heat stability compared to AB and as compared to AA. So beta-lactoglobulin BB is having the highest heat stability. Now earlier as we can see that alpha S1 is having 4 genetic variants here shown, alpha S2 4 variants, and beta casein has having 7 variants, that here we A1, A2, A3, B, C, D, E, and also kappa casein is having 2 genetic variants. So the as when we are discovering the new variants, these are being added into the list. So probably now there are more number of genetic variants are there. So let us now see an important example of A1 and the genetic polymorphism. So we must have heard a lot of times that about the A1 milk and A2 milk. So A1 milk is obtained from cows of Western origin. So they may be Jersey or Holstein flies. we have green sea. So some foreign breeds are there. If milk from those breeds is considered to be as A1 milk, and these cows yield large quantities of milk. So milk production is higher in these breeds, and A1 milk contains beta casomorphin 7. So when digestion of milk occur in the intestine, so there is production of beta casomorphin 7, which is the reason for morphine line effects on the central nervous system. So BCF7 cause addiction of milk, therefore children may prefer to have the milk all the time. So it causes addiction, and morphine effects may cause constipation also in the children. And as compared to A2 milk, lactose tolerance is also high in A1 milk. So there are only few studies which have shown that A1 milk is having some harmful effects to humans, and this has also been reported that A1 milk can cause epidemiological evidences are there, but these are very small. So that A1 milk is reported to cause type 1 diabetes, coronary heart disease, arteriosclerosis, sudden infant death syndrome, autism, schizophrenia. So some of the diseases which have been reported, those studies are very low, and it cannot be said that A1 milk is better or A2 milk is better. But in media hype is there, and then hype is going on. Maybe the marketing gimmicks or not, that we don't know, but there are few reports, few articles are there which says that A1 milk is a risk factor for type 1 diabetes, coronary heart disease, arteriosclerosis, sudden infant death syndrome, autism, schizophrenia. Okay, now if you see the A2 milk is obtained from Indian origin cows like Sahiwal, Gir, Haryana, that other Ongol and so many Indian breeds are there. So if milk is from these breeds is considered to be as A2 milk, and these cows may have relatively less milk, so they give there's less production of milk from them, and they do not produce BCM7 during digestion in the intestine. So A2 milk increases better immunity in humans. This has few reports have indicated that A2 milk increases better immunity in humans, and all those effects which have been shown for A1 milk are not there for A2 milk. And there's some minor studies, or the few studies are also there which indicate that A2 milk is better than the A1 milk, but as such we cannot say, because no scientific data or more studies to be carried out to firmly say that yes, A1 milk is more than the other. Otherwise, if you see what is the difference between A1 milk and A2 milk, that we have a protein beta casein in milk, and in A1 beta casein at 67th position. Here this is 67th position. We have histidine in A1. We see that histidine is there in A1, and this provides a weak bone, and when digestion occurs, BCM7 releases in the intestine. Whereas for A2 milk we have proline at the 61st position, which is provides a strong bone, and there is no formation of beta casomorphin 7 during the digestion. So this is the major difference chemistry wise or genetic polymorphism wise. So this example of genetic polymorphism. So I've told you that these two type of milk differs in the amino acid sequence. Or amino acid only one amino acid at the 67th position. We have histidine in case of A1, and A1 beta casein, and proline is there in the case of A2 beta casein. And there are no scientific evidences are there, and more studies to be carried out to say that one milk is more better than the other milk, and otherwise milk is as such is a complete food, we say that. So here we can see that A1 milk comes from Bos taurus, that is Holstein, Price, Curran, Swiss, Jersey, Brown Swiss. Milk from these animals is considered as A1 milk, and A2 milk Bos indicus, that is Gir, Sahiwal, Kankrej, Red Sindhi, and more Indian breeds are there, Haryana, Ongol. So from milk from all these breeds is considered to be as A2 milk. So this is the example, the trendy example of genetic polymorphism. Thank you.